Chapter 3 Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker in 1969, wherein he suggested the 5 kingdom classification: Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So the cyanobacteria, that are also referred to as blue-green algae, are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms under Plantae. Let us also look at classification within angiosperms to understand some of the concerns that influenced the classification systems. The earliest systems of classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves, etc. They were based mainly on vegetative characters or on the androsium structures (system given by Linnaeus). Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial systems gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment, as against these natural classification systems developed which were based on natural affinities among the organisms and consider not only the external features but also internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of the characters can be considered. Cytotaxonomy, that is based on the cytological information like chromosome numbers, structures, behavior, and chemotaxonomy, that uses the chemical constituents of the plant to resolve confusions, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll bearing simple thyroid autotrophic and largely aquatic (both freshwater and marine) organisms. They occur in a variety of other habitats, moist stones, soils, and wood. Some of them also occur in association with fungi, lichens, and animals. Example on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like Volvax and filamentous forms like Eulothrix and Spirogera. Figure 3.1. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated motile and on germination gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix or non-flagellated non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as an isogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous. Example Volvex and Fucus. Figure 3.1 Algae. A. Green algae. First Volvex. Second Eulothrix. B. Brown algae. First Laminaria. Second Fucus and third Dictyota. C. Red algae. First Porifera. Second Polysiphonia. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on the earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds, which form the basis of the food cycles of all aquatic animals. Many species of Porifera, Laminaria and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produced large amount of hydrocolloids water holding substances, example algin brown algae and carrageen red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella, a unicellular algae rich in proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. 3.1.1 Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made up of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in Zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous or oogamous. Some commonly found green algae are Chlamydomonas, Volvex, Eulothrix, Spirogyra and Chara (figure 3.1a). 3.1.2 Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms ectocarpus to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of the xanthophyll pigments fucoxanthin present in them. Food is stored as complex carbohydrates which may be in the form of laminarian or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contain, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe, and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous or oogamous. Union of gametes may take place in water or within the oogonium (oogamous species). The gametes are pyriform (pear-shaped) and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum and Fucus (figure 3.1b). 3.1.3 Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigments or phycoerythrin in their body. Majority of the red algae are marine with greater concentration found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization development. The common members are Polysiphonia porifera (figure 3.1c), Gracilaria and Gelidium. Table 3.1 Divisions of algae and their main characteristics. Classes: Chlorophyceae, Phaeophyceae, Rhodophyceae. Common name: green algae, brown algae and red algae. Major pigments in Chlorophyceae: chlorophyll A and B. Major pigments in Phaeophyceae: chlorophyll A, C and fucoxanthin. Major pigments in Rhodophyceae: chlorophyll A, D and phycoerythrin. Stored food in Chlorophyceae: starch, in Phaeophyceae: mannitol and laminarian, and in Rhodophyceae: Floridian starch. Cell wall: in Chlorophyceae: cellulose, in Phaeophyceae: cellulose and algin, in Rhodophyceae: cellulose, pectin and polysulfate esters. Flagellar number and position of insertion in Chlorophyceae: 2, 8, equal, apical. In Phaeophyceae: 2, unequal, lateral. In Rhodophyceae: absent. Habitat: in Chlorophyceae: fresh water, brackish water, salt water. In Phaeophyceae: fresh water (rare), brackish water, salt water. In Rhodophyceae: fresh water (some), brackish water, salt water (most). 3.2 Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Figure 3.2. Figure 3.2 Bryophytes. A liverwort: Mercansia. A. Female thallus. B. Male thallus. Mosses: C. Funaria gametophyte and sporophyte. D. Sphagnum gametophyte. Diagram of female thallus: this is Archegoniophore. Gamma cup: this is rhizoids. Diagram of male thallus: this is Anthridiophore. This is gamma cup and rhizoids. Diagram of Funaria: gametophyte, sporophyte, capsule, seta, leaves. and this is main axis and rhizoids. Diagram of sphagnum. This is antheridial branch and this is branches and archegonial branch. Bryophytes are also called amphibians of plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid and shaded localities. They play an important role in plant succession on bare rocks / soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack two roots, stems or leaves. They may possess root-like, leaf-like or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence it is called a gametophyte. The sex organ in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate antherozoites. The female sex organ called archegonium is flask-shaped and produces a single egg. The antherozoites are released into water where they come in contact with archegonium. An antherozoite fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses along with lichens are the first organisms to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverwort and mosses. 3.2.1 Liverworts. The liverworts grow usually in moist sandy habitats such as bank of streams, marshy ground, damp soil, bark of trees and deep in the woods. The plant body of a liverwort is thalloid, example Mercansia. The thallus is dorsiventral and closely appressed to the substrate. The leafy members have a tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structures called gamma (singular gamma). Gammae are green multicellular asexual buds which develop in a small receptacle called gamma cups located on the thalli. The gammae become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into a food seta and capsule. After meiosis, spores are produced within the capsules. These spores germinate to form free-living gametophytes. 3.2.2 Mosses. The predominant stage of the life cycle of a moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping, green, branched and frequently filamentous stage. The second stage is the leafy stage. which develops from secondary protonema as a lateral bud. They consist of upright cylinder axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bear sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs Antheridia and Archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops in a sporophyte consisting of a fruit, seta and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum and Sphagnum. In bracket figure 3.2. 3.3. Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purpose and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, sandy places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes the main plant body is a sporophyte, which is differentiated into two roots, stem and leaves. In bracket figure 3.3. These organs possess well differentiated vascular tissues. The leaves in Pteridophyta are small in bracket microphyls as in Selengenella or large in bracket macrophyls as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones in bracket Selengenella equestum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophyte called prothallus. These gametophytes require cool, damp, sandy places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called Antheridia and Archegonia respectively. Water is required for transfer of antherozoites, the male gametes released from the Antheridia to the mouth of Archegonium. Fusion of male gametes with the egg present in the Archegonium result in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are similar kinds. Such plants are called homosporous. Genera like Selengenella and Salvinia, which produces two kinds of spores, macro in bracket large and micro in bracket small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophyte respectively. The female gametophyte in these plants are retained on the parent sporophytes for variable periods. The development of zygote into young embryo take place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Coelopsida in bracket Coelotum, Lycopsida in bracket Selengenella and Lycopodium, Sphenopsida in bracket Equistum, and Pteropsida in bracket Diopteris teres adiantum. Figure 3.3 Pteridophytes: A. Selengenella, B. Equistum, C. Fons, and D. Salvinia. This is diagram of Selengenella. This is stem, leaves, roots. This is diagram of Equistum, strobilus, node and internode, and this is branch. This is diagram of Fons and this is diagram of Salvinia. 3.4 Gymnosperms. The gymnosperm in bracket gymnos, naked sperma seeds, are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. In bracket figure 3.4, one of the gymnosperms, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza in bracket Pinus, while in some others in bracket Cycas, small specialized roots called coralloid roots are associated with nitrogen fixing cyanobacteria. The stems are unbranched in bracket Cycas or branched in bracket Pinus cedrus. The leaves may be simple or compound. In Cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extreme of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form legs or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspore develops into a male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophytic is called a pollen grains. The development of pollen grains takes place within the microsporangia. The cones bearing megasporophyll with ovule or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be borne on the same tree in bracket Pinus. However, in Cycas, male cones and megasporophyll are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucleus. The nucleus is protected by envelopes and the composite structure is called an ovule. The ovule are borne on megasporophylls, which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms the male and female gametophytes do not have an independent free living existence. They remain within the sporangia, retained on the sporophytes. The pollen grains is released from the microsporangium. They are carried in an air current and come in contact with the opening of the ovule borne on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharge their contents near the mouth of archegonia. Following fertilization, zygote develops into an embryo and ovules into seeds. These seeds are not covered. 3.5 Angiosperms. Unlike the gymnosperms where the ovules are naked, in the angiosperms or flowering plants, the pollen grains and ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest Wolffia to tall trees of eucalyptus in bracket over 100 meters. They provide us with food, fodder, fuel, medicines and several other commercial important products. They are divided into two classes, the dicotyledons and the monocotyledons. In bracket figure 3.5, the dicotyledons are characterized by seed having two cotyledons, reticulate venation in leaves and tetramerous or pentamerous flowers, that is having 4 or 5 members in each floral whorls. The monocotyledons on the other hand are characterized by single cotyledonous seeds, parallel venation in leaves and trimerous flowers having 3 members in each floral whorls. The male sex organs in the flower is the stamens. Each stamen consists of a cylinder filament with an anther at the tip. Within the anthers, the pollen mother cells divide by meiosis to produce microspores, which matures into pollen grains. The female sex organ in a flower is the pistil. Pistil consists of a swollen ovary at its base, a long cylinder style and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form the embryo sac. Each embryo sac has a 3-celled egg apparatus, 1 egg cell and 2 synergids, 3 antipodal cells and 2 polar nuclei. Figure 3.5 Angiosperms, A. Dicotyledon and B. Monocotyledon. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grains after dispersal from the anthers are carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tube grows through the tissue of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where 2 male gametes are discharged. One of the male gametes fuse with the egg cell in bracket syngamy to form a zygote. The other male gamete fuses with the diploid secondary nucleus to produce the triple primary endosperm nucleus in bracket pen. Because of the occurrence of the two divisions, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo in bracket with 1 or 2 cotyledons and the PEN develops into endosperm, which provides nourishment to the developing embryo. The synergids and antipodals degenerate after fertilization. During these events, the ovules develops into seeds and ovaries develop into fruit. The life cycle of an angiosperm is shown in figure 3.6. Figure 3.6 Life cycle of an angiosperm. 3.6 Plant life cycles and alternation of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies.
haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophyte plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following patterns. 1. Sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plant is the free-living gametophyte. This kind of life cycle is termed as haplointic. Many algae such as Volvox, Spirogyra and some species of Chlamydomonas represent this pattern in bracket figure 3.7a. 2. On the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few-celled haploid gametophyte. This kind of the life cycle is termed as diplointic. An alga fucus sap represents this pattern in bracket figure 3.7b. In addition, all seed-bearing plants, that is gymnosperms and angiosperms, follow this pattern with some variations wherein the gametophytic phase is few to multi-celled. 3. Bryophytes and pteridophytes interestingly exhibit an intermediate condition, haplodiplointic. Both phases are multicellular. However, they differ in their dominant phases. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte, and it alternates with the short-lived multicellular sporophyte totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular sporophytic autotropic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplointic life cycle. All pteridophytes exhibit this pattern in bracket figure 3.7c. Interestingly, while most algal genera are haplointic, some of them such as Ectocarpus, Polysiphonia, Kelps are haplodiplointic. Fucus and alga is diplointic. Figure 3.7 Life cycle pattern: A. Haplointic, B. Diplointic, C. Haplodiplointic. Summary: Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperm and angiosperms. Algae are chlorophyll bearing simple thalloid autotropic and largely aquatic organisms. Depending on the type of pigments possessed and the type of stored food, algae are classified into three classes, namely chlorophyceae, phaeophyceae and rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes, which may show isogamy, anisogamy or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright cylinder axis bearing spirally arranged leaves. The main plant body of a bryophytes is gamete producing and is called as gametophyte. It bears the male sex organs called antheridia and female sex organs called archegonia. The male and female gametes produced fuse to form zygote, which produces a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant body is a sporophyte, which is differentiated into two roots, stems and leaves. These organs possess well differentiated vascular tissues. The sporophytes bear sporangia, which produce spores. The spores germinate to form gametophytes, which require cool, damp places to grow. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of male gametes to archegonium, whereas zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are the plants in which ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed and therefore these plants are called naked seeded plants. The gymnosperms produce microspores and megaspores, which are produced in microsporangia and megasporangia borne on the sporophylls. The sporophylls, microsporophylls and megasporophylls are arranged spirally on axis to form male and female cones respectively. The pollen grains germinate and pollen tube release the male gamete into ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovules into seeds. In angiosperms, the male sex organ (stamen) and female sex organs (pistil) are born in a flower. Each stamen consists of a filament and an anther. The anther produces pollen grains (male gametophyte) after meiosis. The pistil consists of an ovary enclosing one to many ovules. Within the ovule is a female gametophyte or embryo sac which contains the egg cell. The pollen tubes enter the embryo sac where two male gametes are discharged. One male gamete fuses with egg cell (syngamy) and other fuses with diploid secondary nucleus (triple fusion). This phenomena of two fusions is called double fertilization and is unique to angiosperms. The angiosperms are divided into two classes, dicotyledons and the monocotyledons. During the life cycle of any sexually reproducing plant, there is alternation of generations between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns of life cycle, haploid, diploid and intermediate. Chapter end. Thank you for watching and subscribe my YouTube channel.